So I will start with the intro. Thanks everyone for coming to another COVID seminar. Just a reminder, we have 2 more for the rest of the year, next Friday and the following Friday. Today I'm very excited to have Dr. Song Lee with us. Song is the Chancellor Professor and Chair of Department of Bioengineering. He's also the Professor of Department of Medicine. Song got his PhD and postdoctoral training in bioengineering at UCSD. He became the professor of bioengineering at UC Berkeley between 2001 and 2015, where he also served as the co-chair of Berkeley UCSF graduate program and co-director of Master in Translational Medicine program. In 2016, Song got recruited to the bioengineering department at UCLA. So really, Song is really loves the UC system. He's been going all along the California coast. Dr. Lee's research is focused on cell engineering, biomaterials, drug delivery, mechanobiology, and regenerative engineering. And his lab has been taking an interdisciplinary approach to engineer stem cells and micro nanomaterials, which really advances the understanding of how biophysical forces, factors regulate cell reprogramming and tissue remodeling and informs therapeutic design. He's filed over 10 patent applications, which really shows the translational potential of his work. And he's very accomplished and has been elected as a fellow of American Institute of Medical and Biological Engineering, Biomedical Engineering Society, and the International Academy of Medical and Biological Engineering. It's a mouthful. We're really excited to hear his recent efforts to apply some of this work to COVID-19. Please take it away, Dr. Lee. Thank you.
Thank you very much, Melody, for the kind introduction. It's a great pleasure to be here. Really, this is my first time to talk about this research, and I got most of this data actually in the last week. I think this is just kind of internal seminars. I'd like to share some of our recent work. At the same time, hope to get some comments and input from the audience, and all these scientists here. So first, I want to have a brief overview of what's going on in bioengineering. And really, just in response to this COVID-19 pandemic, a lot of things going on in our department, and many of our faculty have adopted technologies in trying to address these, a lot of these challenges in diagnosis and therapeutics. So for example, regarding device, and some of our faculty work on the PPE design and 3D printing, making a point-of-care device, and some of them work on a novel imaging method trying to facilitate the diagnosis of COVID-19. And many of us in the molecular and cellular engineering area, we work on drug delivery, vaccine delivery, and also potentially use some of approach in tissue engineering for the regeneration of various tissue and organ. And in addition, there are also some basic research ongoing to use AI and data science to explore some of the disease mechanisms. So here's one example done by Jake Schmidt. What he did was to use all these 3D printers to print the PPE in different kind of design. And at the beginning of the pandemic, that really helped to make the first batch of these PPEs for healthcare workers. And Ben Wu actually did much more work afterwards, and they call this PPE 2. And then actually it's a website on dentistry. You can see a lot of detailed information there. And they did a lot of work to produced a prototype of many different things, including the PPEs and some ventilator, valves, splitters, etc. So I don't know any of you had a chance to attend the bioengineering seminar yesterday. In this quarter, we have a COVID-19 seminar series. And on the one hand, we want to educate our students about various technologies that can be used to address the challenges in this pandemic. In addition, we hope to have a kind of research showcase what's going on in our department. So here is one example for point of care diagnosis. In Kamei's lab, what I'm trying to do is to use the system. They did this before for other kind of diagnosis applications. And there's also a startup to further develop this for COVID-19 diagnosis. So basically here, they're trying to use the two-phase separation to concentrate the samples and use this lateral flow immunoassay. This is about an inch size device. It's very similar to the pregnancy test chip. And you can use this to detect several things from the blood sample or saliva samples. And they have developed assays for several different kind of detections. RNA from SARS-CoV-2, they could detect the antibodies in the blood samples, and also the SARS-CoV-2 proteins. So based on what he said, it seems this detection is closer to the finish line and to be a product in the near future. In DiCarlo's lab, what I'm trying to do is to use this microsphere device to deliver vaccine with some kind of a drug delivery capability, incorporate some of those adjuvant into this microsphere to enhance the vaccine efficacy. There's some also research going on in drugs. One's lab is trying to do to use machine learning technology to study the protein structure and also try to address various fundamental questions regarding COVID-19. For example, the cytokine storms. How can you suppress this antiviral defense in the immune system, etc. So now I want to just focus on what we are doing in our own lab. So many of these results actually are quite preliminary. It's all unpublished. So in our lab, we focus on cell and tissue engineering. On the one hand, we are trying to understand how microenvironmental factors, including the biochemical factors and biophysical factors, together regulate cell functions, especially stem cell differentiation. And recently, we focus on a cell reprogramming model. In addition, we have moved on to study how immune cells can be regulated by various microenvironmental factors. Our interest currently is really on the T memory stem cells. In addition, we are interested in just translate what we learn in this basic research and use that for various tissue regeneration applications. We have done quite some work in the area of blood vessel regeneration and neuromuscular regeneration. And now we are trying to address is whether we could use some of this approach, especially in the drug delivery area, to enhance the T memory stem cell function. So you all know that there are a lot of progress in vaccine development recently, and especially for example, mRNA-based vaccines. Of course, there are many other kinds of vaccine in the pipeline. For example, inactivated virus, this is already in the phase 3 clinical trial, and also peptide-based vaccine as well. So while these vaccines all have, some of them will have some side effects, but at least the mRNA vaccine demonstrate very good protection with over 90% of efficacy. So, but there are some issues, for example, for the delivery of this, shipping, storage, and delivery of the mRNA-based vaccine, and that could be a problem. So there's some space for us to do something, for example, to increase stability of this vaccine in the future that will help us to be able to deliver large number of these vaccines to the remote place in countrysides. In addition, for now, let's say if we give vaccine, we have 2 shots, and there's a possibility we can use drug delivery approach to just give 1 shot, and that will decrease the amount of work clinically by 50%. And with the drug delivery approach, we can allow a sustained delivery of the signal, or even have a control release of 2 very strong signals at let's say 2 weeks interval to reach that. So delivery, that's definitely something we can do to enhance the vaccine for the prevention of disease. The other thing we are trying to do is to address this issue of immunosenescence. So we know the senior people or this aging population are highly vulnerable to this disease. They can easily get an infection, and also the immune system actually basically decline with age, and that's a big problem. And many of them, if you talk about the flu vaccine, every year, many of the senior people may not respond very well to these vaccines. So one big issue is the long-term memory responsible for these T cells and B cells. In addition, even for the current vaccine, because we're trying to rush to get everyone a shot to stop this pandemic, and we haven't got long-term really efficacy data at this time. So we don't know whether this vaccine will be able to maintain a long-term memory. And there are some cases, for example, people may get a second infection. So whether this is a specific for this virus, whether the immune system in response to this specific vaccine could achieve a long-term memory for our immune system, is not very clear. So for delivery, here's just one slide to show some of the things we're trying to adopt to use for vaccine delivery. And we use this for immunomodulation in the periodontal disease. In this case, we use these biodegradable microneedles. And here, this microneedle, you can see there are 2 parts. This needle is biodegradable. You can tailor the degradation rate. So we can release different kind of molecules, peptides, proteins, small molecules. In the back support, this membrane could also be degradable. In this case, it could melt within minutes to hours, and the rest of these microneedles will stay in the tissue and allow a sustained release of material. So we have used this for as a patch for periodontal disease. In this case, what we're trying to do is to modulate the immune response in the infection area. When we have this issue here, for example, the pro-inflammatory macrophages will be responsible for the damage and degeneration of periodontal tissue. And if you could convert this into the pro-regenerative type of macrophages, and that will be helpful. So we're using this microneedle patch for periodontal disease treatment. And we are also interested in using this for the vaccine delivery. Actually, a capstone team is working with our postdoc and grad students, undergrads in our lab, trying to develop this microneedle system for vaccine delivery. So, okay, now we want to talk about some of this recent work on T memory stem cells. So T memory stem cells was discovered in the past 2 decades, in more and more evidence showing that this type of cells plays a very important role for the long-term memory of our immune system. So if we have a naive T cell, upon stimulation, of course, the cells will differentiate, and some become the effective T cells. In a very small percentage of this could become quiescent and remain as memory T cells. And many of those are memory T memory stem cells. So these cells can self-renew and maintain a long-term memory. And of course, it's dependent on a certain type of virus or disease or bacteria. In some case, the people have shown that, for example, for the yellow fever virus-specific T memory stem cells, it could be detected even after 2 decades. So that shows a very potent response in long-term memory. So to address this issue of in the aging population, in this case, they have naive T cell, but the formation of T memory stem cells is retarded. So to enhance that, we could do a few things to promote the formation of T memory stem cells. So here are 3 things we are working on. These 3 approaches. One is to make artificial antigen presenting cells as a vaccine booster. So in this case, you can use these artificial APCs to deliver various signals to promote naive T cell activation, and also trying to arrest them at the T memory stem cell state instead of further differentiation. And a second approach, which is slightly different, even though we have signal on the surface or inside of this microsphere or an artificial APC, we could do is, in addition, to generate an artificial niche in the body to fulfill a part of function of a lymph node. So in this case, we can include a synthetic signal in this scaffold that can be injected into the subcutaneous space and or near the lymph node, and then we could use that to modulate immune cell response locally together with a vaccine. In the third approach, what we could do is take advantage of the native antigen presenting cells. In this case, what we could do is have some kind of drug delivery system to load all of these signals to antigen presenting cells. And when these antigen presenting cells migrate to lymph node, they could release all of these payload, these drugs locally in the lymph node to modulate the T memory stem cell formation. So here we have most of the recent work focusing on the first approach. So I will just show some of these preliminary data. So antigen presenting cell or artificial antigen presenting cell is not a new concept. It has been used for cancer therapy, for example, for CAR T therapy, for the ex vivo expansion of the T cells or activation of T cells. This has been widely used. And one commercially available example is Dynabeads, right? So on the Dynabeads surface, you could code it with antibody as an activation signal. CD3 antibody or CD28 is commonly used. You can use that to activate T cells. Of course, Dynabeads has a lot of issues or drawback. One is this is a kind of a stiff material, and which is quite different from antigen presenting cells. So as bioengineers, what we could do is to engineer the material itself with different mechanical property or stiffness, or even make it very similar to the real antigen presenting cells, and to facilitate T cell activation. In addition, we could also add more signals to these antigen presenting cells. So here, in our approach, we just choose a vaccine candidate. In this case, it's a spike protein peptides. And this is a commercially available. There's a library of peptides from spike protein. Of course, if any vaccines come into clinical use, we could use that together with our vaccine booster. So here is a kind of an overview of what we are working on. Let's say you have a naive T cell here, and we first is to do the same thing as others have done to activate, to have this activation signal on the surface, including the anti-CD3 antibody and anti-CD28. In addition, We have interleukin-2, which is just kind of background signal to help activate the T cells. And there are some other things specific for T memory stem cells. One is this GSK3 inhibitor. And this inhibitor could activate a Wnt pathway. And Wnt pathways has been shown to play a very important role for T memory stem cell formation. In addition, for the renewal of these cells and expansion, interleukin-15 and interleukin-7. So of course, our goal is also to simplify this design. For example, whether we need all of these cocktail or we just need a part of them, and we're trying to figure it out as well. So here is what we expect, how it could work in vivo. And let's say if you have these artificial APCs, and these are microspheres, and we can inject them subcutaneously. And we have all these signals carried by these microspheres. So this signal, let's say we could deliver that with vaccine candidates, could be peptides or mRNA vaccine, and whatever people have figured out. And a couple of possibilities. One is that the antigen-presenting cells can take this signal, and then they, for example, the spike protein peptides, and take them to the draining lymph node and activate the T cells and B cells over there. In addition, if we have this, let's say the resident T naive T cells, or even the progenitors of T memory stem cells in the local tissue, and we could use this antigen presenting cell as to fulfill part of function of all of the signals in the lymph node. We could activate these cells and promote formation of T memory stem cells locally. Of course, these cells could also go into the lymph node and finally to activate B cells, etc. So here's the fabrication system we use to make microspheres. So here we have a microfluidic system. We can adjust the flow rate of this sheath fluid or the fluid in the middle. And then we can control the size of the particles we make. So this is some examples of certain size of particle we have made here. In the previously, actually, Marty Hassani is a scientist in the lab. He really spearheaded this project and did some previous work with Manish Butte and several other faculty on campus to try to make, in this case, it's artificial T cell. Of course, you can mimic the size of the cells, and you could also code it with different molecules on these microspheres. And this is a mechanical characterization showing that the mechanical property and size can all be controlled. And here shows some of these artificial T cell interact with B cells in culture. So here is a basic characterization. If you have, let's say, different density of surface activation signal, or different sizes, or even different mechanical property of this microsphere, and you could examine the effect on T cell activation. So here, in this case, the material for microsphere is alginate, which is the major component of seaweeds in biocompatible. So for microsphere, we include different components in this drug delivery system. So in the microsphere, we have several things to deliver. One is the chemical. The chemical is hydrophobic, so we basically load them into the PLGA, polylactic glycolic acid nanoparticles, and load these nanoparticles in the microsphere. So this can be controlled for sustained release. In addition, for all of these cytokines or any other growth factors, we take advantage of their affinity binding to heparin. They have some heparin binding domain. So we conjugate heparin to the alginate backbone and can load all of these cytokines with a different amount, and the release can be slowed down compared to direct mixing of these cytokines into the microsphere. So here shows the control release profile as an example. One is cytokine, for example, interleukin-2. You can see we have a quick release at the beginning, and there's a sustained release for about a week or so. And in addition, for chemical small molecule, this GSK3 inhibitor, we also have a sustained release. So all of this release profile can be tailored in a way as we want. For example, if you want to have 2 weeks release or have a slow release, we can adjust the polymer or other chemistry conditions to achieve that. So this is an overview of our experiment design. And what we're trying to address now is the difference in young and aged animals in the mouse model. So our goal is to demonstrate that, let's say in young animal, probably all of this immune activation is normal, but in aged animal, if you have decline of immune response, you can boost this T memory stem cells. But of course, we are also interested in address the difference in male and female mice, but we haven't done much in that area yet. In addition, we got this small funding from CERN. We are supposed to address some of this translational potential in human cells. We also did some experiments in a human peripheral blood mononuclear cell as well. So first, I want to show some data in mouse. So in mouse, what we did was to isolate the naive T cells from the spleen of the mouse. You can see here, it's just histology showing the abundance of these cells. And to examine the T memory stem cell differentiation, we take advantage of some known transcriptional factor and also the molecule expression. So we know, for example, naive T cell, if they differentiate in the central memory or effector memory cells or further differentiate into the T cells, in this signal, these molecules, TCF7 and LEF1, will go down. And then if you could just interfere with this differentiation pathway, trying to arrest some of the cells at the T memory stem cell stage, and that would be great. So that's the goal. So if you have some, if our delivery system have some effect, what we expect to see is there's some early peak of this, or there's a delay of decline of this expression of TCF7 and LEF1. Here shows some of these results. So we sort the CD8 positive T cells. And then we have several control group. One is no treatment. We also use Dynabeads as a control as well. And then in this case, the Dynabeads plus the soluble factor, this is the TWS119, which is this GSK inhibitor. And we could also add other soluble signal for the T memory stem cell survival and expansion, IL-15, IL-7, etc. So here you can see that when you have this GSK3 inhibitor, really it increase. There's an early increase of the TCF7, indicating the inhibition or suppression of the differentiation process at early stage. And then when you have this additional signal, we don't see much of a change here, but we need to further determine whether really there's any effect for the expansion of this population. So now if you look at, of course, this for LEF1, it's very similar. And if you look at our artificial APC, you can see here The effect actually is even better. Instead, we're seeing this kind of a decline after a couple of days, and we have kind of a plateau response. So with this chemical inhibitor, you can induce the T memory stem cells, and this could be further enhanced by IL-7 and IL-15. And similarly, for LEF1, we have the same trend. So the other characteristic of the T memory stem cells is that they could be quiescent, and then without, if they're not exposed to the antigen again, they will not be activated. And then in that case, they maintain as a stem cell. So we can use this CFSE dye to label the cells and look at the cell division. When we have these artificial APCs with this chemical inhibitor, you can see that this peak is the cells before when we loaded the dye, and you can see that the division of these cells is slowed down. So to quantify the formation of these T memory stem cells, and we use these markers, CD62L and plus positive, and CD44 negative, and SCAR1 positive as a marker to characterize this cell population. If you take these CD8 positive naive T cells, you can see here, if you have dynabeads with this chemical inhibitor, there's some increase of this population, right? Dynabeads alone with only CD3 and CD28 antibody will not have much effect. If you take our artificial APC, you can see here, just APC alone, even though you add this T cells survival expansion signal, it doesn't matter. You need this specific GSK3 inhibitor to boost this formation of T memory stem cells. So for this early stage of formation of TMSCs, really we think this chemical seems to play a more important role than interleukin 7 and 15. So to characterize these cells, we also use some gene expression analysis. So we know that, for example, for the self-renewing memory T cells, there's high level of interleukin 2 expression and low level expression of interferon gamma. So to see whether indeed we can promote this cell population and decrease this one, you can see here when we add inhibitor with dynabeads, we could see the chemical inhibitor indeed suppress the interferon gamma expression. And for our artificial APC, we have a very similar effect to boost the interleukin 2 and then also suppress the interferon gamma expression in these cells. So now the question is, all of these experiments have been done in vitro, in the absence of the specific antigen. If you add, let's say, any antigen from the SARS-CoV-2 virus, what will happen? So here we did this experiment by adding the peptides to our microsphere. You can see this adding of adding peptides here did not affect the formation of these T memory stem cells stimulated by our drug delivery system. Of course, now the cells will be slightly different. CD122 is induced when you have this antigen-specific CD8 positive T cell formation. And the other question we are trying to address is whether this is true in human. The same kind of cocktail, same drug delivery system. So in human, actually the markers, surface markers for these T memory stem cells are slightly different. And what we did also is to isolate the cells from peripheral blood instead of a spleen. And here is a very straightforward isolation. You use this gradient centrifugation. We can isolate this cell population for further analysis. So indeed, in this case, if you look at all of these effects induced by the artificial APC in our system, we have very similar response as we have observed in mouse cells. With this chemical inhibitor together with artificial APC, we could induce the formation of T memory stem cells. So based on what we have observed, it seems to be this case, when we have this GSK3 inhibitor, and we have an increase of T cells, and when you have these two cytokines in IL-7 and IL-15, even though they are not essential for this early activation, they can further boost the response. For the T memory stem cell, and with the presence of this chemical inhibitor, we could induce this early peak and then kind of suppress the further differentiation of these T cells. We also did some preliminary work in animal model. I want to just show you some of these preliminary data. So when we inject this drug delivery, all of these microspheres into the tissue, we're supposed to examine a few things. One is the humoral response, and the other one is a cellular response. So for humoral response, there are several things, right? So we can first examine the IgM formation at the early stage, and maybe within a week, and then IgG formation probably after 2-3 weeks. So what we did was to see whether this could be a very specific response. We took advantage of this Elisa Analysis. If you have spike protein coated on a dish, and then we could detect whether we have a spike protein specific IgM formation or IgG formation. Of course, there are some other analyses, and we haven't done all of those yet to look into the detail of IgG subclasses, and also look into some other neutralization assay to see whether this antibody is sufficient to neutralize the binding of, for example, virus, or even the real SARS-CoV-2 virus to the cells, and also the entry into the cell. So here is a preliminary data we did observe, and in this case, when we inject into a mouse model, we could induce IgM formation. In this case, we add a few commonly used adjuvant here, aluminum, right? Commonly used for hepatitis vaccines, in poly-IC for cancer vaccines, and also CPG, which is being explored broadly for various immune cell activation. So in this case, in the presence of this spike protein peptide, you can see there's an increase of IgM in all these cases. And similarly, for IgG, you can see that with the adjuvant and also APC, you can see a very similar response here. So we do have humoral response. That means there's some signal going to the lymph node to activate the B cells. For cellular response, there are a few things we can look into. Of course, to directly examine whether the signal goes to lymph node, we can examine the draining lymph node swelling, stiffening, and that's a very common response to vaccine and also any other foreign antigen. And that could be used as a marker for the activation of the immune cells. We can also look at T cell response directly. So here is what happened if you just do not if you do not have any vaccine, right? So let's say you have some kind of adjuvant, you're going to have early activation, right? So just a direct injection. And if you have an artificial APC, we basically prolong this response. In here, we show that even though at the beginning it's not as strong as direct injection, but if you have a sustained release of this signal, you can see actually this response sustained for a couple of weeks. And if you have this chemical inhibitor to induce T memory stem cell, and the response is very similar. And you can look at this time course of lymph node stiffness change. So we also did some experiment to see whether there's difference in young and aged mice. In this case, aged mice is over 1 year old, 1.5 year old. You can see here in young mice, whenever you do here, you give them some adjuvant and also this APC together with this chemical inhibitor, we all have a very strong response, right? So we do not know whether it is necessary to do so in young animals. But if you compare that with the aged animal, you can see some big difference. In aged animal, the immune response is not as strong. But when you have this AAPC, you could enhance this response in with the chemical inhibitor in presence. There could be further enhancement. We need to increase the sample size to see whether this is significant. So here, really, I just show some of our preliminary study, and I hope to get your input. At the same time, I want to thank all the lab members, and Marty is the one leading the project, and also Yana and Sher, who help perform some experiments. Also our collaborators, Manish Butte and John Tsai. Yunshen is a biologist lab, and now Yuan Si is involved in some of these projects. Thank you very much.
Great, thank you so much, Song. That was a really great talk. I would like to open the floor for any questions from the audience. You can just unmute yourself and or show your face. You can also type your questions in the chat box. Maybe I'll start with the first question, Song. I thought it was really cool. Thanks for showing your mouse model data. It's very exciting. Is there any data, I guess, from previous study when approaches like this are used in humans? I guess I'm curious about in terms of variability between individuals, how that could affect sort of the response with sort of this engineered APCs and the time release of the cytokines, and also thinking about how that could affect sort of the inflammatory response, because with COVID, obviously that's a big concern, right, with inflammation.
That's a good question. Actually, I don't think this has been used in any of the clinical application yet. The only thing similar is the one in CAR T therapy outside the body, right? So CAR T therapy, of course, it's a very expensive therapy. It costs hundreds of thousands of dollars for each procedure. And for this application, we don't know yet, but there's also a potential to use that for in situ T-cell activation for cancer therapy. The question is whether this could be potent enough to stimulate the T-cells in the body. If this cocktail could work, and that could also help to improve enhance the memory of T cells. And let's say if some patient has tumor removed, and this memory can help the patient to prevent the further, just the tumor growth again in the body. Yeah, but anyway, so I think it needs to be tested. Even for in vitro study, we need to increase sample size and see whether the donors, it's a different kind of gender or insanity, and all of this, see whether the response are similar or not. So just to see whether, so all of this need to be done. But in animal model, at least, it's encouraging. We observe some enhancement in aged mice. So that's a good thing we can, yeah.
Right. No, that's really cool, because that potentially would be translational into humans in terms of the age differences, I guess. Great. Is there any question from the audience? I guess today our audience is very quiet. Well, if anybody has any, I guess, comments or suggestions for our speaker today, feel free to email Dr. Lee. And thank you so much for your talk and sharing your preliminary data with us. It's great to have you. Thanks so much, Sung. All right. Bye, everyone. See you next Friday.